will do in my part of the presentation is briefly give you the background of metagenomics and kind of following some of the resources that you will also find in the introductory materials on the Learn portal for anyone who would like to kind of get familiar with these concepts before we start the program. I want to kind of start from the very beginning and talk a little bit about, why the microbiome and how exactly can we study the microbiome. So since the discovery of the microscope, we've been learning about microorganisms that surround us and even live inside us. And with the increasing resolution, so here we have a chart of the different types of small things that you can see in the microscope. You can see that the optical microscopy resolution allows us to see kind of all the way to bacteria. And because of that, this kind of interest in the way that the microorganisms live together has evolved as the resolution allowed us to see smaller and smaller things. So what you can see here on the bottom is a figure. This is an image from a microscope, a super high resolution microscope, where you can see E. coli and some phages. So these are viruses that are sitting on the E. coli bacteria. So what has become clear with the increasing resolution is that not only that there are trillions of microorganisms around us, they also live in communities and they interact with each other, not just with their hosts or the environment. So because of that, early on, there has been an acknowledgement of the role that the microbes play in human health and well-being. And early on, you can see here in the mid-1600s, there was the first observation of microscopic organism. And by the 1800s, there has been an association made between disease and microorganisms. But today, in the last couple of decades, there has been a tremendous increase in research concerning the role of the microbiome in a variety of different conditions that affect the environment, but also affect us as people, including different aspects of human health and development of the healthy microbiome on the skin, in the gut, and in other organs. And what we will talk about today are several examples, just to kind of understand what are some of the considerations, how is the microbiome studied, what are some technologies that have been used to do that, how these technologies are evolving, and what we will cover in the program to be able to handle such data practically and kind of understand the insights that we can gain from such data. So one important factor about the microbiome is the stability. So essentially, these communities of bacteria, specifically we'll talk more about the bacteria rather than just the whole microbiome, we can kind of all relate to the stability of these communities when we think about such microbiome communities like our skin. So for example, microbiome on the skin will create a biofilm, and this biofilm can actually communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confers benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. And quorum sensing activities are also involved in the virulence and pathogenic potential of bacteria. And so they're an important factor in understanding and controlling bacteria and bacterial infections, as they enable microorganisms and biofilms to become more tolerant of host defenses and antimicrobial agents. And inside each community, there's going to be a balance of bacteria, fungi, and viruses that control species abundance in skin. So if you touch your own skin, you will feel that there is more than just one type of feeling on your skin, right? Some parts of your skin might be dry, some of them might be oily, some of them might feel smooth or might have bumps on them. So all of this means that there's this whole microbiome interacting with the host. And one question that we might have is, well, what factors are actually determining some of the qualities of this microbiome? How are these distinct communities different from each other? Because we know that all of our skins are probably very different from each other, but also how do the things that we can control affect these communities? And so to answer some of these questions, there has been a tremendous interest in understanding, characterization, and investigation of the factors that really control the microbial communities in many different organs. So you can see here on the screen some of those defined microbiomes, like the microbiome of the skin, the respiratory system, the gut, the intestines, the stomach, the urogenital tract. So all of these, since 2010, I believe there have been hundreds of thousands of papers related to the microbiome and major projects that have been developed. So some of the major projects that you can see right here. In 2010, we have the Human Microbiome Project that we will talk a lot about in this program, but also throughout the years, there have been distinct projects focused on the gut, skin, lung, and even brain microbiomes. So many of these projects, like the Human Microbiome Project, seek to characterize the healthy microbiome because there's a lot of variation, as we already discussed and we'll talk about in a few slides, related to the age, to the location, to the diet and lifestyle, as well as the environment of the healthy microbiome. But then also an additional set of projects have been designed to address some of the questions around disease. So you can see here cancer prevention study for microbiome in cancer. You can see the Alzheimer's gut microbiome project, the nurses health study, and others. And so really, we can see here a variety of disciplines that will be affected by the microbiome, from healthy nutrition and lifestyle, all the way to a variety of precision medicine applications. And even you can see here on the bottom, space travel, right? So it appears to also play a role in space travel and the health of astronauts. And so many different areas of application for metagenomics and for microbiome. So because of all of these projects, we know today that the human microbial communities contribute to many critical and essential metabolic, physiological, and immunological functions. And they include differentiation and maturation of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosal barrier function, development and regulation of the immune system, prevention of invasion and growth of disease-promoting microorganisms, and several others. And so to study these conditions and this variability, there are several different types of omics technologies to study the microbiome. And these could be distinguished into sequencing-based or mass spectrometry-based. So for sequencing, we can look at the DNA-based approaches. So essentially, we can sequence specific genes or try to assemble whole genomes of bacteria or whatever species of these microorganisms. We can also look at RNA-based. So we can try to infer function. What are some of the genes that are being expressed? And what do they mean? What is their function? There are also ways to study the microbiome using mass spectrometry, where we can look at proteins and metabolites that they produce. But today, in this session, we'll talk a little bit more in detail about metagenomic sequencing. And so metagenomic sequencing, metagenomics in general could be characterized as both a field and a specific technique to study the microbiome, but it is different from many other types of omics. So essentially, many types of omics are focused on characterizing a single system, a single individual. For example, when we speak about the genomic studies, we're trying to understand the genome of an individual. Metagenomics, in that sense, is different because it looks at a community. So specifically, when we speak about a community, we think of the microbiome as almost like an organ that has a specific function. And one of the ways that we want to understand the function of this organ is by capturing the diversity of microorganism within that microbiome or microbiota, the community that it forms. And when we do that, it is different from a lab-based technique where essentially a single microorganism is isolated and is grown in a different environment than where it actually typically lives. And so because only 1 to 2% of bacteria can be cultured in the lab, metagenomics allows us to study how these microorganisms coexist together with others and respond together to different environmental changes or produce some functional effects. So the difference between these two, we can see here for example, when we want to study the gut microbiome, one of the ways that we can use sequencing to understand this microbial community is by trying to annotate the different taxonomic variation found within the gut. And sometimes we can also combine that with an understanding of a particular individual representative of that community to try to understand what does that specific individual bacteria, for example, how is it different from others, right? So we'll talk a little bit about how these techniques could be applied to different types of conditions in just a moment, but let's try to see how this type of information could be generated and what are some of the techniques to then study the data that is being generated from sequencing. So two kind of approaches that I want to discuss here. The first one is sequence-based, when we are able to sequence all of the DNA or the RNA that is present in a sample, like next generation sequencing. So this could be shotgun metagenomics, 16S amplicon metagenomics, or metatranscriptomics. And then sometimes we also have these approaches with qPCR when we know that we're looking for specific types of bacteria, and we could design primers to actually find whether there is an abundance of a particular type of a bacteria or combinations of bacteria. So if you think about the next few slides, you'll see how both of these could probably be applied to answer similar questions, but we'll more speak about next generation sequencing because of the richness of information that it can provide us with, and how we can apply qPCR, for example, to validate the findings that we will see in next generation sequencing. So really, shotgun metagenomics or metatranscriptomics differs from 16S amplicon metagenomics in the way that we're able to capture the specific view of the composition of the microbial community using 16S amplicon metagenomics, or essentially thinking about the 16S ribosomal RNA as a marker gene that helps us understand taxonomy. And then shotgun metagenomics or metatranscriptomics is the analysis of all of the genes within an ecosystem. So the difference is, are we sequencing all of the genes or all of the genome, or are we looking at conserved marker genes or gene regions? And some of the challenges that are associated with both of these approaches. So first of all, in trying to sequence all of the data that is present in the DNA that we extract from this microbial community, obviously assembly is going to be one of the major challenges. How do you take these short reads and try to piece them together to extend them into an assembled genome, or at least a part of the genome that then we can search for specific genes and try to annotate them. So even with 16S amplicon sequencing, we have this challenge of annotation as well, because essentially not all of the bacterial genomes are known, But because we have databases that are constantly being updated, because of these major studies, it is possible to define the taxonomy or the diversity and the presence or absence of specific types of bacteria in a given sample. So let's kind of talk about 16S metagenomic sequencing and try to understand the specific applications of 16S metagenomic sequencing, which I think are very exciting in the context of human health and well-being, different types of disease applications, and even behavior, as we will see. So 16S amplicon sequencing is a technique that is widely used, one of the most kind of widely used techniques. And this sequencing focuses on the 16S ribosomal RNA gene. So first, that gene is amplified with PCR amplification, and that gene contains hypervariable regions that allow us to study taxonomy of a particular community. So one of the challenges with the actual sequencing is first to organize the sequences that we get into what we call operational taxonomic units. And these are units that resemble each other with some specific markers. And then we can use those to annotate and try to understand what do they match in a database. And so there are several different databases like the Silva, Green Genes, there's the NCBI repository of all of the bacterial genomes, and as well as RDP. So what do these databases essentially provide us with? So these databases can help us understand taxonomy, or if we think about the hierarchy of biological classification, how we can study the more or less detailed view of the different types of organisms. So the taxonomy is just a way to organize the different types of organisms into some kind of a structure so we can see what is more related or less related. So starting from the bottom here, kingdom, phylum, class, order, family, genus, and all the way to species. Now, as you will see, not all of these are going to be available for all of the individual microbial organisms that we will have. And so what we can try to assess are the differences at maybe specific levels, like for example, class, phylum, or order, or sometimes dig deeper into the species level of classification. So to really understand how diverse the microbiome is and how many organisms we're dealing with, let's actually look at the Silva database, and we'll see there how many different organisms you are going to be finding there. So I'm going to paste the link in the chat box, and I will actually address your questions here in a few minutes. So please go with me to this link. Okay, so this is the database, the Silva database. Here you will find the different levels So right here, you can see those levels: domain, phylum, family, major clade, class, genus, kingdom, and order. And so you can see here, bacteria, right? That's the kingdom. And then it goes all the way down to family right here. So what you can do here is you can search by sequences, by accession number, or by full name. And here you will be able to find by taxon name, or you can also browse here specific types of bacteria. So as we go through, I will give a couple of examples of bacteria that you will be able to find here and search for it right here in the taxon name, and then you'll be able to find it. Okay. Beyond the bacteria, right, what we need to remember is that a microbiome contains also viruses, as well as other microorganisms. So because we have this interaction between bacteria, the community that it forms, and other microorganisms, right? We won't talk about it as much today, but you have to remember that the same types of databases exist for other microorganisms that you might expect to find in the microbiome. So again, viruses, for example, as well as you can see here, the virome, right? So in the same way that we can call the microbiome all of the microorganisms, we can also look at specific types of microorganisms. So for example, for viruses as well, called the virome.